Hello and welcome to today's TAS webinar, Antibodies to Watch in a Pandemic. It is now my pleasure to turn the floor over to Dr. Janice Reichert, Executive Director of the Antibody Society. Janice, when you're ready, the floor is all yours.
Thank you very much. Welcome everyone to Antibodies to Watch in a Pandemic, and thank you for joining us. As was mentioned, I'm Jan Reichert, Executive Director of the Antibody Society. I'll give my presentation first, then I'll introduce our guest speaker, Dr. Thomas Sherman. He has been incredibly busy lately, directing efforts to discover anti-SARS-CoV-2 antibodies with his UMAB and Karat Therapeutics colleagues. We are delighted he made time to be here with us today. First on the agenda is my update on antibodies to watch in 2020, which will cover antibody therapeutics that have been approved so far this year, and also antibody therapeutics that may be approved by the end of the year. I'll then provide an overview of antibody-based COVID-19 interventions, including repurposed therapeutics that treat symptoms, and newly developed anti-SARS-CoV-2 antibodies, which I consider the stars of today's show. Dr. Sherman will then discuss his experience with the accelerated development of anti-SARS-CoV-2 antibodies via an academic industrial consortium. We'll then have time for questions. Our first topic is recent first approvals in the US or EU and the projection for 2020. This graph shows the number of antibody therapeutics approved in each year in either the US or EU. As you can see by the height of the bars on the right-hand side of the graph, the biopharmaceutical industry has done remarkably well at getting new antibody therapeutics through the development process recently. Since 2014, at least 6 new antibodies were approved each year, which can be attributed to the involvement, of course, of more companies, but also improvements in technologies that have allowed more antibodies to enter clinical study. You'll notice that the projection for 2020 indicates an extraordinarily high number of approvals. So let's go through the specifics having to do with those antibodies. 5 antibody therapeutics have been approved so far in 2020. All were approved in the US, with only 1 approved so far in the EU. They are for a variety of indications, including thyroid eye disease, migraine prevention, multiple myeloma. We had an approval for an ADC for triple-negative breast cancer, as well as approval recently, just June 11th, for an anti-CD19 molecule for the treatment of neuromyelitis optica spectrum disorder. With only 5 approved, you might be wondering how we're going to get to 20 approvals by the end of the year. The answer lies with the 16 antibodies currently in US or EU regulatory review. Now, those of you who are good at math have already noticed 5 plus 16 equals 21. In reality, 1 of the molecules has entered regulatory review recently, and I don't think the timing will work out for a 2020 approval. For the 16, I've ordered the molecules as best I can by the time of an anticipated approval. In the US, it's very helpful to know the PDUFA dates, which are the dates by which FDA is supposed to take a first action on the application. 3 antibodies have PDUFA dates in August and July. For the first antibody, the ADC for multiple myeloma, just really recently in the past day or so, it was announced that the Oncology Drugs Advisory Committee to FDA will meet on July 14th. So we'll hear on July 14th what those advisors think of the data for that molecule, which has a PDUFA date in late July. Satalizumab, which is an anti-IL-6 receptor molecule being evaluated as a treatment for neuromyelitis optica spectrum disorder, has a PDUFA date in late August. I'll also be mentioning this one, of course, in the context of COVID-19. We also have an anti-CD19 molecule for diffuse large B-cell lymphoma with a PDUFA date in late August. An additional 5 have PDUFA dates known to occur by the end of the year. Again, here you see antibodies for a variety of indications. All of these have either U.S. breakthrough therapy or fast track designations, indicating they address a currently unmet medical need. Regeneron's mixture of antibodies for Ebola has a PDUFA date in October. I will, of course, talk about their mixture of anti-SARS-CoV-2 antibodies that has entered clinical studies recently in the next section. Meeting these PDUFA dates does depend on the availability of FDA resources. So far, they're doing quite well at meeting existing review deadlines while also dealing with this extra workload associated with COVID-19. If all goes well, and it does seem to be on the right track, we may have antibodies approved by the end of the year for Ebola, cold agglutinin disease, neuroblastoma, breast cancer, as well as osteoarthritis pain. Besides the 8 antibodies in review that I've shown so far, there's an additional 7 in U.S. review, but I don't know the PDUFA dates for these at this time. FDA does not release the information, and the sponsoring company may or may not release the information. PDUFA dates can be estimated, though, based on the date of the BLA submission, if the company's mentioned that, as well as the likelihood of a priority review. The 5 shown here are for non-cancer indications. You'll notice they all have fast track or breakthrough therapy designations. A benefit of these designations is priority review, which has a 6-month time frame for the first review cycle. Since these are all in review already, and they likely have a priority review, it follows that they may be approved by the end of the year if the first action is an approval, which is very common actually. But I want to throw a caveat out there. Please do bear in mind, I'm telling you what may happen, not necessarily what will happen. Here we have a couple of more antibodies in review with unknown PDUFA dates. These are for cancer, including an immunotoxin for bladder cancer and dostarlimab for recurrent microsatellite instability high tumors. The antibodies that I've mentioned so far are the 15, with a chance, at least in my estimation, for approval by the end of the year. The other molecule is tralokinumab, an anti-IL-13 molecule for atopic dermatitis that entered review in the European Union very recently, and I think an approval in 2021 is more likely than 2020. For the most part, antibodies entering review do get approved. It's rare for an antibody not to be granted an approval, although sometimes it does take more than one review cycle. So that wraps up my update on the non-COVID interventions, the non-COVID antibodies to watch. Summary from that section is so far so good. Cautiously optimistic that we will indeed have a record number of antibodies approved in 2020 despite COVID-19. However, we are in the midst of a pandemic, and despite improvement in some regions of the world, no one can relax because SARS-CoV-2 is not going away. We need drugs, we need vaccines, and we need a lot of them. Like many organizations, the Antibody Society did a hard pivot toward COVID-19 topics in March. Initially, we started with the post-series, Coronavirus in the Crosshairs, which examines ongoing discovery and development of drugs and vaccines, as well as diagnostics. We also started collecting publicly disclosed data on the biologics, and reached out to the Chinese Antibody Society for their help because, quite frankly, there was a lot of content coming out. Some of it coming from China, and I myself do not read or speak Chinese, so their help was very, very important in understanding what's going on in China. This collaboration led to development of a tracker, which is a searchable online database, and I will be referring to that tracker multiple times in the rest of the presentation. We're now tracking approximately 130 antibody-based COVID-19 interventions that are sponsored by commercial firms. Of these, approximately 35% are repurposed biologics being evaluated for disease symptoms, and approximately 65% target the virus itself. The data we have is from the public domain. We do not think it represents all ongoing efforts, as of course, not all companies will discuss their preclinical work. The tracker includes approximately a dozen-ish programs that to date are sponsored solely by academic or government organizations. For the purpose of this presentation, I've excluded them because those types of organizations typically do not gain authorization to distribute drugs. This is usually done by companies. So my assumption is that promising candidates from academic or government sources will be licensed by a company, and if that happens, I'll fold it into future analyses. So now I'll talk about the therapies that are for symptoms. As you are likely well aware by now, there are a lot of medical conditions associated with COVID-19. The infection by the virus causes an acute respiratory distress syndrome initiated by cytokines. This can lead to pneumonia. It can lead to tissue damage resulting from hyperinflammation. COVID-19 patients have also been observed to have abnormal clotting. With so many disease-associated pathways involved here, of course, people are familiar with antigens that are, or proteins that are involved in these pathways, and they are indeed targets for over 40 antibody-based therapeutics that were already in development. Relevant targets here include the interleukins 1, 6, and 8, as well as GM-CSF. Of the antibodies in development for COVID-19 symptoms, 9 target IL-6 or the receptor, 6 target GM-CSF or the receptor, and 4 target C5, or it may be described as C5a, or the receptor. However, there are many less frequent targets. Targets are presented here in alphabetical order to make it easier to find your favorite one. You'll see many are associated with inflammation. For example, the more interleukins are shown here. I would not take the time to go through all of these today, but I invite you to go to the tracker to find more details about these antibodies. With the time remaining for this section, I want to focus on the antibodies that have advanced the furthest and thus may be eligible for authorization soon. In this graph, the green bars represent the most advanced phase of development, and the blue bars represent the most advanced phase specifically for COVID-19. You see 11 are approved, with one of these approved for COVID-19. The other 10 are either in phase 2 or 3 for the disease. Our current count shows 21 at phase 2 and 16 at phase 3. So I want to note that some of those phase 2s may be pivotal. That is, the data may result directly in some type of an emergency use authorization without any need for a phase 3 study. So everything in phase 2 and phase 3 certainly I would count as antibodies to watch. So I'll now discuss the approved molecule and some of the antibodies at phase 3. The authorized antibody therapy is a human mAb targeting IL-6 receptor that was developed by BioCAD. It was developed originally for rheumatoid arthritis, but BioCAD started a phase 3 study in April, late April even, included over 200 participants who received a single dose. According to their press release, the results of the study demonstrate that the antibody therapy can significantly reduce mortality among patients with COVID-19. On the basis of the study results, BioCAD was able to register the molecule on June 5th. So this is incredibly fast, with Russia having also some type of a fast-track mechanism for registration of molecules for COVID-19. 6 of the molecules in phase 3 were already approved for some other type of indication. Tocilizumab was an obvious choice as a drug for COVID symptoms because it's already approved for a type of severe life-threatening cytokine release syndrome. It is included in over 55 clinical studies of COVID-19 patients, with 3 of these sponsored by Genentech Roche. These are relatively large studies with primary completion dates between July 31st and August 31st. Positive results could allow authorization as a COVID-19 therapy. 2 other previously approved antibodies targeting IL-6 or the receptor are in late-stage studies. Sarilumab is being evaluated in 2 phase 3 studies sponsored by Sanofi or Regeneron. One has a primary completion date in July. The second is very large, involving some 2,500 patients at 65 study sites, but the primary completion date is in May 2020. However, as I've been saying, positive early results might allow an earlier authorization. So Cetuximab is also in a phase 3 study, but this is not sponsored by a commercial firm. An additional 3 antibodies approved for other indications are also being evaluated in late-stage studies in COVID-19 patients. Targets include interferon gamma, interleukin-1 beta, and C5. In addition, leronlimab, which is in regulatory review, FDA review, for HIV infection, is also in a phase 3 study for COVID-19. So where does all of this lead? Assuming there are positive results from these studies, then the sponsor can approach FDA for an emergency use authorization. In late March, FDA was given the authority to allow use of unapproved medical products or unapproved uses of approved medical products, which is what we're talking about here, for COVID-19. However, I do want to point out that there are limitations to the scope and the duration of such authorizations, and there may be specific conditions that apply to these authorizations. I also want to point out that there may be some confusion that I've seen here or there about different phrases that are used, such as emergency investigational new drug applications or compassionate use of a COVID-19 intervention. That is not the same as an emergency use authorization. To date, to the best of my knowledge, an emergency use authorization has not been given, it's not been issued for a biologic, at least not yet. And an emergency use authorization, just to be very clear, is not the same as an FDA approval. So I will consistently be referring to these as authorizations for their use, not approvals. Sponsoring organizations, of course, may not necessarily pursue an FDA emergency use authorization, but presumably they would be pursuing some similar type of a mechanism through their home country's regulatory authority. Now I'm going to move on to the stars of the show, the anti-SARS-CoV-2 antibodies. The tracker includes over 80 anti-SARS-CoV-2 biologics. There's a lot of content in there. Again, I invite you to go have a look for specifics about the molecules that I'm talking about. The majority are traditional mAb-based therapeutics, including nanobodies. About 12% of them are some other type of composition of matter, including derpin, Fc fusion protein. It's one that's nucleic acid-based, coding for an antibody. There's also some polyclonals that are recombinant or transgenic animal-derived. Of the antibodies, the monoclonal antibodies, where we have known derivations, about half of them come from B cells of convalescent patients, with about a quarter each from in vitro methods such as phage display or immunization of animals. I do want to remark, though, that not included in those numbers are some organizations that have already stated they're using 2 or even 3 of these methods. So you've got a lot of resources. You might be going through multiple of these approaches. For the molecules that we're tracking, not surprisingly, most of them are in discovery or preclinical. For the purposes of the presentation and also in the tracker, we put molecules in the bin or programs in the bin of preclinical if the company or organization was talking about data that they have on protection in animal models, or they were developing their cell lines, or they're preparing for clinical studies. If what was discussed was simply screening or assessment of neutralization, then it's in the discovery bin. A total of 6 have proceeded, though, into clinical studies, with 4 of these being antibodies, which are the ones that I will focus on next. 2 of the molecules are in phase 1 already. You know, it seems like really, really quick progress. It is the case that 2 organizations have already gotten their molecules into phase 2, or in the case of Regeneron, they refer to their studies as phase 1, 2, 3. Both could generate pivotal data that could possibly support an emergency use authorization. Regeneron's is a mixture of 2 different antibodies, and the primary completion dates for the studies are in November and in March of 2020. Lilly's, with their partner Abcelera, they have a molecule that's in phase 1 as well as a phase 2. Those 2 studies have primary completion dates in August and September. So again, early study results might result in an earlier authorization, which potentially could happen as early as September. Numerous other organizations have announced plans to transition anti-SARS-CoV-2 antibodies into the clinic by the end of the year. Details are included in the Coronavirus in the Crosshairs, Part 9 of the series, as well as the tracker. These organizations include UMAB Corat Therapeutics, of course, and we'll be hearing about their efforts very soon. It also includes Celltrion, Vir Biotechnology, Sorrento, AstraZeneca with their partner Vanderbilt University Medical Center. Which I have to remark has also other antibodies that they're partnering with other organizations. Distributed Bio has been very active and has a molecule that they hope to get into the clinic by the end of the year, as does Verna Therapeutics with their partner University of Toronto, Shanghai Junshi Biosciences, as well as Molecular Partners with the derpin molecule. We also want to mention some of the companies that have not as yet announced plans for a clinical study, but they have programs that may progress by the end of the year, and we await some type of an announcement of such from Twist Bioscience, Pacifica, Iontys with their partner FairJourney Biologics, Immunoprecise Antibodies, Ligand, Ablexis, as well as fusion antibodies. So it's truly been an extraordinary effort by the biopharma industry, and especially small organizations, to get these antibodies through the process very quickly and into the clinic. So I'll just wrap up with some key messages having to do with the projection in 2020. It really looks like it could be a record year for approvals of non-COVID interventions. provided FDA and other regulatory agencies don't have their resources diverted too badly by the really extensive workload associated with COVID-19. FDA's emergency use authorizations for biologics, I do believe, will be coming along relatively soon, and one of the first ones may be tocilizumab. The quite extraordinary response by many, many organizations located across the globe in developing anti-SARS-CoV-2 antibodies could potentially lead to authorizations of some sort, either from FDA or potentially in a different country, Celltrion, for example, in South Korea. But it could lead to such authorizations for 15 to 20 different antibodies. Again, we need as many of these as possible to be available for patients. First emergency use authorization is possible as early as September based on the studies that are already ongoing. Finally, I'd take just 30 seconds here to thank our corporate sponsors for their support. Logos are arranged alphabetically here. If you see your company's name and you're not registered as a society member, please do go to our website and register because it is free. We have over 30 sponsors: Biocytogen, Distributed Bio, FairJourney, Genmab, Janssen, and Lava Therapeutics joined recently. We thank them, as well as all of our sponsors, for their support, especially during this very challenging year. UMAB in particular has been a very strong supporter of the Society, and it is with great pleasure that I introduce Dr. Tomas Sherman of UMAB and now also Corral Therapeutics. Dr. Sherman received his PhD in immunology for his work in adoptive cell immune therapy with CAR natural killer cells from the Humboldt University, and he worked as scientist and research group leader in research institutions, including the University of Braunschweig. In over 20 years of his research, he gained broad interdisciplinary expertise in recombinant antibody technologies, protein engineering and expression, bi- and multispecific antibodies, fusion proteins, gene-modified immune cells, and immunotherapies. He is the author of more than 70 research articles and inventor of several patents. He's co-founder and CEO of UMAB, a fast-growing biotech company. He's received numerous prizes for his outstanding work in translating science into commercialization. Currently, he also acts as CEO of two UMAB spin-offs, UMAB Incorporated based in the US, which was founded in 2017, and Corral Therapeutics based in Germany, which was founded this year. Dr. Sherman, I turn the webinar over to you.
Thank you, Jan. I think most of the audience is here because of your overview, which is incredible. I'm always enjoying to read your antibodies to watch, and it's always a great pleasure to have you in the conferences. Thank you also for the introduction and for all the organization. I hope that the quality of the audio will be fine, because we sometimes struggle here a little bit. Today I will talk about UMAB's approaches, which finally end up in just a few weeks ago to a spin out to move our COVID-19 fast track antibody project towards to clinical development. But let me give you a very brief introduction. What is UMAB doing? You can already imagine we have something to do with antibodies. So we are a German company spin out from the University of Braunschweig, a city which is located in the middle of northern Germany, just 2 hours away from Berlin. And we are a privately, mostly CRO company, working for more than 100 clients all over the world and helping them to develop human antibodies and to help them also to engineer antibodies. So we are a small company, so we are among those who try to help with an anti-COVID-19 antibody therapeutic, such as other companies in the world. And yeah, I also want to mention here, especially our co-founders from UMAP, which are also shown here on this slide, which is, except me, is Andre, Stefan, and Michael. And especially Stefan and Michael were, I think, the first people who tried to form an alliance between academic and industrial experts, which they also already called CORAD, just in the beginning of March, I think. And then we were joining with UMAP to support their efforts. As you also can see, UMAP is not the only company in CORAD. There are also other spin-outs from Stefan and Michael. So there are a lot of things going on. And yeah, let me just very briefly also show what UMAP is doing actually. When I say we develop antibodies, this is not just a discovery. It's also associated with a lot of different technologies which have to work tightly together. And our main focus is here to develop antibodies from the target to a final lead, which in principle is ready for the next development steps. Antibodies are mostly fully human, natural sequence based, close to germline with our naive human antibody libraries. We have a very fast in vitro discovery approach, and we can address also very different types of targets and develop different antibody formats. Yeah, so this is actually just to show that our help and contract research supports big projects for our clients. So it's of course not only our job here, but we're very happy that we from time to time help our clients to move a project and to give them the opportunity to make a big deal. So then, you know, everything could go on for the next years and everything is fine. But then 2020, when I think I was in January sitting in San Francisco and then later in San Diego with some colleagues together, we heard, okay, there's a lung disease popping up in China. We didn't really think it will change the world actually. However, when you look today, and this is just a screenshot from Sunday evening, yeah, just a few days ago, we were already confirmed more than 10 million infected people, which is probably just a little cut out of the true number, which we actually don't know, yeah, especially when we are telling people we don't need diagnostic tests. Yeah? And also what we can see, there are more than 500,000 deaths officially related to COVID-19. Yeah. So this picture really depicts, like from a teaching book, how a pandemic should look like. And I think we have never experienced anything after the Second World War like this. And more importantly, we don't have a vaccine, we have no curative medicine, so somehow the only way to do is we try to preserve the life as long as possible until patients' own immunity somehow gets rid of the virus. This is what we have today. Fortunately, there are some developments ongoing, and we are very hopeful also as UMAP and CORAD Therapeutics that we may have some part in it in the near future. So this is the timeline of the pandemic. When you, I don't go into the details, but it's not very long ago. I mean, when everything changed for us in our way how we live our lives nowadays, and you can see UMAP was probably one of the first companies, at least I'm aware of, who's involved in antibody generation with our in vitro technologies, and we already finished our first contract research project with Boehringer Ingelheim by the end of March. Meanwhile, Stefan and Michael, who I've shown you already, they already started with academic collaborations, trying to do their own project, and then later a lot of people joined this consortium, which they called CORAD. And later you see that it's the same name of the new company. We joined them and started a second approach, Of course, we first talked to Boehringer that there's no interference with their project, and we get our first virus-neutralizing antibodies by the end of April. And already the first discussions to get some scientific advice with our regulatory authorities. It's the Paul-Ehrlich Institute in Germany. For a fast-track development approach, which has not been done so far, at least in Germany. We get a very positive response later, and we stuck there somehow because we could move on and get into the clinic if everything works well by the end of the year, but we didn't have any funding. And that's when you see the limitation of academic research. If you're running out of funding, you somehow cannot do anything anymore. And that's why we decided, okay, UMAP has a good understanding how to spin out companies in the past. So let's form a new company, put all the IP into the company, and then move on. And just a few weeks later, we already had our first financing round to move on, and now we are very, very briefly before the DP0 stages, which is to initiate cell line development and every other step. And hopefully by the end of the year, we may be able to initiate the first clinical studies. I'm not going to talk too much about the SARS virus. I think the SARS coronavirus 2 is I think well described, and you all know about it. And Janice already started to talk a little bit about it, so I skipped this slide for the sake of time. Importantly, not everyone who gets infected has a severe outcome. A lot of people get very slight symptoms, but even they are not protected by another infection, as we see today. A lot of people do not develop antibodies or not sufficiently, and we don't know so much about the virus, which is actually a big problem, because if the biology is not known, the biology is always the driver of developing a drug or a vaccine. So that means we have a lot of unknown questions here. However, what we know is it's extremely transmitted from human to human and now spread almost on every place on the world. So we are not secure anywhere else. And the severe outcome is often a reaction, maybe not directly related to the virus itself. Of course, it's caused by the virus somehow, but it's often an overshooting inflammation and cytokine storms, which can result in a lot of other symptoms leading to organ failure and death. And when you look now on this next slide, you can see this is just data from Germany and one data set from Italy. As you can see, the risk groups where are really in danger to die from COVID-19 are alone in Germany, it could be 15 to 20 million people. And when you see the death rate, death toll, just related to the age, you can see between Italy and Germany, it looks very similar. When you get more than, if you're older than 60 years old, it dramatically increases the mortality. And with 70 years, almost a quarter or 30% of the people can die. Yeah, and this is, of course, in industrialized countries, with a lot of older people, a big problem, and especially when it comes to limitations in the infrastructure, with the intensive healthcare units, the death toll can rise dramatically, as we unfortunately have seen in different countries, even in very, very well-developed countries. So we have 4 strategies. I'm not going to all of them today. Just to mention, a lockdown and quarantines are not really useful for long-term perspective. They help. They definitely help, yeah, and they probably saved a lot of infections and deaths, which could happen in Germany. So we were really lucky, at least until now. But it does not, it cannot be done for always. So if we shut down everything, the economic loss goes into trillions of dollars, and it's clear that it's not an option for long term. Diagnostics are very important because they give us an idea how many people are infected. Unfortunately, we do not test everyone. Either it's too expensive and a lot of people are asymptomatic and still can infect other people. So we need all of these structures, all of these strategies, and including, of course, vaccines. But vaccines are not a cure, so they provide protection for those who are successfully vaccinated and get a good protection. However, to get a herd immunity, we need to vaccinate up to 60 to 70 people. And you can imagine this takes many years. I don't know if it's ever possible, and it will cost tens of billions of US dollars. So the last strategy, of course, is therapy and drugs. And as you understand, this is not about the drug itself. It's the whole complex of how to treat a patient. You need intensive care units as well as life-saving treatment opportunities, and we need them today and not in 3 years. Yeah, and antibodies are one of these strategies among the antiviral treatments. However, I also should mention, we have seen already from Janice that we also need drugs to cure or to reduce some of the severe symptoms, like anti-inflammatory drugs, including antibodies. There's always a question if there's a race between a racing duel between vaccines and drugs. Just to compare again, it's very simple overview. We need both of them, but vaccines just protect if you immunize and vaccinate millions of people. The advantage of drugs is if you once have a sufficiently effective and safe drug, you can just focus on those who are in danger with a severe COVID-19. And at the end, the costs will be probably similar or less for the drugs. And if you have a drug today available, you can immediately save lives. If you have a vaccine available today, you still have not saved too many lives because you still have to vaccinate billions of people. So just to conclude this slide, drugs are as important as vaccines, and maybe even more urgently. So just also to take a little comment on the fact that there are hundreds of drug programs on the way to treat COVID-19. We need these programs, yeah. And realistically, even from a phase 1 program, normally 10 programs, maybe one will get into a market-approved medical drug, and that means you need 10 programs for one drug. If you have 100 programs, you may have 10 drugs, but at the end, we need as many as possible because we have to address different mode of actions. The virus may mutate and new strains arise, and maybe one of the drugs loses efficacy. Then there's different clinical stages which may require different types of drugs. Also, we have to address different symptoms where maybe a neutralizing antibody against COVID-19 is not sufficient. And that's why a combinatory therapy is relatively realistic in the future, which we require, at least for the late-stage patients. And the big problem here for a company, independent, if it's a big or small company, is if there is no market, usually industry would make a step back. I'm very happy that this hasn't happened so often yet, but I can tell you from my own experience when talking to potential partners, the response was always like, okay, we need to look first on the market, and unfortunately, no one really can predict what will happen in 1 or 2 years. We can see what's happening today, and we have unfortunately also the proof that the second and the third and whatever number of waves are coming up in the future will happen because it's already happening. Yeah, and the other problem here is that the market is not only unpredictable; it's very unpredictable for a company if this is profitable business. So you need some vision, and you need to understand at least that it's important to do something, yeah. And of course, when the market is uncertain, the governments are in the role of supporting us. I have to say, so far in Germany, we didn't have much luck about it, and we also need the combination of scientists because the biology is. I mean, I'm very happy that a lot of publications has been done very rapidly and helped us to understand the disease better, but we also know that's not the end and we still have to learn about it. And starting from an academic consortium, at the end, we also know industry has to take over at least to supply the market with the number of doses required. Yeah, drug development as we know it from a teaching book, it takes 10 to 15 years and costs $1 to $3 billion US. We understand, first of all, this is not available for every company, and second of all, we cannot wait 10 to 15 years. At least then we are all dead or we are already immune. Yeah, so that means we have to address high risks in reducing the costs and also accelerate the development. And you can do it in each of the steps. I just summarized it here. At the end, it's clear you need robust platform and a clear mechanism of action, a clear vision. Of course, the proof will be in the clinic first. And you need to generate a lot of different antibodies, not just the one. Of course, at the end, you have one lead, but you have to address other things than only neutralization of a virus, but it has to be also developable and manufacturable. Then you also have to include potential safety measures, because if you fail in phase 1, you have to go back to step 0 and start again, and you lose a lot of valuable time. Probably this program will fail at this stage. And at the end, of course, efficacy is supported by a fast assessment, and again, a clear mechanism of action is important, because if you don't know or if you don't believe in the biology, the mechanism you want to target on the virus, then of course it's very uncertain if you will ever get into the clinic very soon, because you also have to convince the regulatory authority that this will work. So this is actually already a little bit better understand for vaccines, because there were past epidemics with similar viruses like SARS and MERS or Ebola. And what we have learned out of it, that the old strategy didn't work out so well, because at the end, the vaccines were never available before the epidemic already has ended. And this is what we cannot do anymore. We cannot wait 3 years or longer to have a vaccine available. And we have already learned that they have learned to do things faster, and that there's a lot of concepts already in the clinic, and maybe we have a vaccine relatively soon. Important in this new concepts is that they have been based on better preparedness from those companies, and also a nested and parallelized development strategy. So you start much earlier with the manufacturing, even if you don't know if you will be successful. And also there's a certain kind of public funding already available for vaccines, at least in Germany. What I've learned recently, there's a lot of more money. I think 750 million euro has been invested just to support the manufacturing of vaccines. So I think we have to learn from that and take over some of these steps and of course adjust them to drug development. And this is what we believe we need, all the expertise we get. We need a certain kind of pragmatism, because we cannot make a perfect drug in a very short time. So we have to look what is safe, what is efficacy. Perfection is not possible. So we have to give up the idea that this drug maybe will do everything in the future and will cover all the coronavirus in the next 20,000 years. This will not happen, of course. And we need, of course, to include the regulatory authorities, because we do things which have never done before. So we have to discuss what is possible, because we do not want to fail, because we haven't done a good job and we don't get approval for the clinical studies. We need also standardized processes which are well understood, which is very good actually in the antibody field, and we need parallelized workflows. So when in early March, especially you see Stefan and Michael from the University of Braunschweig started to talk to a lot of people if they can do more than just making some antibodies. They joined a lot of people, even from big pharma, and finally we formed several subgroups and up to 50 people involved or more, and to try to develop a fast-track approach to treat COVID-19. And another advantage for us, especially for UMAP, is we are located in one of Germany's infectious disease research hotspots at the science campus in our city, Braunschweig. And we have here the Helmholtz Center, which has security level 3 facilities and animal facilities. We have a GMP manufacturer also just around the corner. And this helped us to move on and coming up with a plan. And this is just a very brief overview how it works. We started to make antibodies as we always do, very fast. We also included, of course, samples from convalescent patients. We developed new antibody libraries. We screened them with our approach. We tested them for virus neutralization and other properties. And we are now very briefly before the DP0 status, meaning one lead moving forward, this one, to develop clinical an IMPD, which is ready and safe for clinical studies. So the mechanism of action I do not have to explain. So what we want to do here is very simple. We want to neutralize the virus from binding to its host receptor, and by this prevent infection. That this approach works, we have already, our co-founders from the university have already demonstrated in many, many different scientific publications up to preclinical work in animals. Even with very nasty viruses like Ebola, it works. However, in coronaviruses, it gets more complicated because coronaviruses are known also to use some mechanisms which we do not really understand so far, that antibodies non-sufficiently neutralizing the virus can enhance infection. This so-called antibody-dependent enhancement can be a serious problem because it guides the virus to other cells which expressing Fc receptors and thereby becoming infected, and can have a very severe outcome, probably by secreting cytokines and other things. And this is also very important because we see in severe COVID-19 already that inflammation is a severe phenomenon which maybe leads to the very bad outcome of the disease. So that's why we, right from the beginning, after some discussions, we decided we need to address this, because safety first is also important, not just to throw something out on the patient and hope it works, but to do not make it worse. Therefore we designed some safety measures into the antibody to prevent an ADE completely. And these are now some data. Sorry, I have to keep things a little bit shorter because of the sake of time. You can see we screened a tremendously amounts of libraries from different patients. We ended up with more than 750 human antibodies, different antibodies, more than 250. I think more of them were neutralizing inhibiting the interaction of the virus receptor binding domain with its host receptor. We finally decided we have to reduce the number somehow, so we have to select some of the antibodies to do the live virus neutralization tests, and later on we did some additional CMC assessment and bioinformatics, and now just I think a few days ago we reduced the number of 3 top candidates and 2 backup candidates, which will be reduced further after tissue cross-reactivity testing and further CMC assessment to 1 lead candidate, and then go on for the cell pool cell line development. So the diversity, this is just a little example out of it. We get very diverse responses from immune libraries. You can see several clusters of antibodies. Finally, we use this to reduce the number of antibodies a little bit, because we believe that each of the similar antibodies will have a similar epitope. So we try to get as many different epitopes, which we still don't know because a lot of these epitopes are conformational, into our next step of the development. At the end, we did virus neutralization, which is shown on this picture. You see on the left side, there's a in red surrounded it's a non-neutralizing antibody, which looks like a normally infected cells. On the right side, you see a neutralizing antibody gets the same result like non-infected cells. So we have 100% neutralization. And this is just another picture of it. You can see the red one is actually the one on the slide before. It's actually from our naive library. So obviously some of our own antibodies may provide already some protection, or one of the donors of our libraries had already neutralizing antibodies. But you can see lots more of the neutralizing antibodies coming from immune libraries, which we would also expect. And this is the status we are today. So we are very soon in DP0 stages. We have subnanomolar virus neutralization of live virus. We have not found yet any developability issues, and hopefully it will be confirmed on a larger scale. We think we have a very safe format of the antibodies to do not raise any security concerns. And we are now going into the next steps. It's a cell-pool cell line development with parallelized steps. So even all the steps we do today we will do in the next future will be different from what we have done ever before in a normal process. So it's not a stepwise process. Things will be done in parallel. We have accelerated preclinical studies and talks, and we will start very soon also an ongoing stability studies, which will not be finished before the IMPD. So this is an enrolling process where we have to follow up from time to time with the regulatory authority. But we believe if we have enough data, this will be no stop to go into clinical testing by the end of the year. So this is my last slide. I want to thank our partners. I'm not sure if we can go over 1 hour, but I also want to mention the students from the University of Braunschweig, which supported the project. So I think they worked many weeks, 7 days a week, and a part of their antibodies are also in the later lead development process. Ended up, we are soon before the next step, DP0, and we hope we will be in human patients by the end of the year. Still, the funding is still relatively tight, even after first financing round. So we are also looking for partners, of course, and maybe for the next generation, which might be more complex, but will need more time. So I thank you for your listening, and I'm opening for questions.
Tomas, thank you so much. Let's take a couple minutes, and we do have a couple questions here. And I'll throw them up to Janice and you here to answer. One of the questions asks, Janice, are you aware of anyone developing antibodies for COVID-2 that target elsewhere on the virus, i.e., not on the spike protein?
I'll also direct that question to Tomas, because I myself can think of only one. It might have been a nanobody where they seemed a little unclear of exactly what the mechanism of action is. For the most part, the ones that we are tracking are to spike protein. Tomas, have you heard of anything else?
Also I cannot, I actually don't have the same overview like you in detail. I think from our rationale and from probably anyone else, it makes sense to go on the spike protein. But I have to comment that on the spike protein, there might be more epitopes. And at the end, these antibodies may work together from different companies. I mean, we have seen there are some approaches with 2 antibodies already on the way. But at the end, each of these approaches may be undergone by escape from the virus by some mutations. So this is a big question at the end. Yeah? So to say I am not aware of any other approach that's really going away from the S protein.
Great. Our next question asks, anyone know how long it takes, anyone know how long it takes from lead antibody sequence to human clinical trial? Virus has publications saying 6 months.
So,
well, Tomas can speak from his experience, but I would say, based on Lilly's public disclosures, that it took a maximum of 4 months for them and potentially closer to 3. Tomas, do you have input on this question?
I can only, for our project, and I have no idea how they made it. Yeah. So we have already a plan to go from the lead, and it took us more effort to find the right lead. So this, I don't know, I cannot comment on the others. About 5 months from the lead to IMPD, which is the potential starting point to go into the clinic. To do it in 3 months is extremely sophisticated, and my only explanation is that the system they had already in place works more tightly together. So we have to work with external companies, for example. As you can imagine, a 25-people company like UMAP and Cohort, which is just founded, cannot do it internally. So my only explanation, they have used already established systems and without thinking straightforward. But I think 3 months is the absolute minimum which is possible. I think you cannot go, except you switch to transient expression, but I'm not aware that anyone is trying to do transient expression to go into the clinic yet.
Awesome.
Yeah, because the stable cell lines take some time. A pool or monoclonal, it takes time, and I think this is a limiting step in the process. And you need to really use standardized procedures, so you cannot optimize any step in it because you would lose much time. Formulation classically can take around 6 months. Imagine only for formulation. So go step, you need 18 to 24 months typically. So.
Great. Let me see here. Our next question asks, does the Antibody Society track combination therapies, specifically for combinations of antibodies approved or in clinical trials? If so, where would I find that information?
There are far too many combinations for us to track combination studies. We do track mixtures, so I would definitely draw that distinction. For example, Regeneron's mixture of the anti-SARS-CoV-2 antibodies, if it's antibodies that are combined together and then evaluated as a product candidate, we would track that, but not independently developed antibodies that are just put together, which is typically what people mean when they say combination therapy. So not combinations, but we do have mixtures. And if you want more information on that, please do feel free to contact me at the Antibody Society.
Yeah, but I think the project from Eli Lilly and AbCellera, there are 2 antibodies too. I've read something about it, which is one is I think from the Chinese company and the other from AbCellera. It's a combination. But this is, I read it yesterday somewhere. If this is really true, I don't know. And this is, again, started already as a combination. So it's a mixture or cocktail or whatever.
Well, I know they went into clinical studies independently, but it does make sense for them to have combined them and to be evaluating them as a cocktail, absolutely.
What I believe is actually, I mean, we don't know what will happen with the pandemic in the future. So let's say in half a year, the virus changes, and when first therapies are successful, maybe it changes somehow, and maybe some of the drugs will not work anymore alone, and then you maybe need the combination, or you need just different drugs. The question is, how many drugs do we need to successfully get rid of this virus? And the answer is no one really knows. Yeah.
Great. Looks like we do have one more question here, and it asks, when you mention selecting a safe format, is this IgG or fragment?
We have a classical IgG format with some changes in the FC to do not elicit any severe or no effector functions. This is the goal. And this has actually also proven in animals already by another group, I think from China, if I'm correct. Sorry, I'm not totally up to date with the scientific publications anymore due to other problems to solve in the company, in the new company. But I think we have already some proof how to do it, and we may have some different angles in the way how we try to do not elicit such unwanted responses.
Great. Well, I don't see any more questions here. Janice or Thomas, any final thoughts before we wrap up here?
Thomas?
Yeah, maybe briefly. What I've learned in the past is that it's not just a biological problem or a technology problem. I was very satisfied about how people, scientists especially, could easily interact in starting a consortium without thinking about agreements and things like this. So how fast we could start a project, and then when we try to put everything into a company, things getting more complicated. So when we're thinking about endeavors like this, we always think it's a technological problem, but it's more than this. It just really needs support by funding, by partner companies and more, and even needs changes in thinking. For example, in the legal, how to make agreements possible in a shorter time. These things really cost time. Fortunately, we were lucky. We could make things in 4 weeks possible, which usually take a year. We didn't do a due diligence, for example. Just people had to believe us, and then we had to go through the legal process. This is very, very fortunate. But I think for the future, we need more preparedness, and we need preparedness on a level which goes beyond technologies. We need also preparedness in the governments. We need preparedness in the big companies. We need preparedness how to put academic scientists, how combine them. And for example, it is always a question at the end, who is inventor of something, how to share the results, because at the end, the world needs to benefit from that. And of course, the company needs to be profitable, but I think we have to really leave our conventional ways of thinking under normal circumstances and give up something to really be faster in the future. Yeah. Janice, what do you think? I mean, you are the goddess of antibodies.
Thank you, Thomas.
Yeah, I know.
I totally agree with you. I think all of the things that you said are completely spot on, and we do need to be better prepared in the future. I do think we have learned a lot. People have been referring to COVID time, that is how fast things have been speeded up, and now we have a better understanding of what actually is possible in a short period of time. Nevertheless, the learning has come at a pretty severe cost, and we do hope we're better prepared next time around. With that, we are reasonably past the hour. I do want to thank everyone for logging in and being with us for this hour. If you have additional follow-up questions, please do feel free to contact me. Those that are for Thomas, I'll pass along. He's busy right now, so I'll act a little bit as the gatekeeper, if you don't mind. And again, thank you very much, and have a good rest of your day.
Yeah, thank you very much for the invitation. Janice, you did a great job again, like usual. Yeah, And thank you for all the participants, which I think we have now 173. I'm very, very impressed how good this works. And maybe we have another update soon, hopefully with more data and more successes.